Hello and welcome to lecture 17 of the lecture learning about learning lecture series. Just before this we have been looking at various evidences of behavior being controlled at different scales, particularly at the level of molecules. We had a proof from dopamine, measuring the dopamine levels and then correlating with the sign related versus a goal related behavior, and then even causally proving that you can actually modulate these levels and then change the animal from one way or the other. And we also looked at the whereabouts of how the protein synthesis, the role of protein synthesis was discovered and how it actually came by. Towards the end we were in looking at the different the neuroanatomical features of the hippocampus, and then how Bliss and Lumo were planning to use those these features to study the plasticity in the brain in the hippocampal region. And there we were talking about how one can actually use the different electrodes to stimulate and to record and study the throughput of the the input output, or what happens to that in response to quote unquote an event. Now we will look at that in more detail in this lecture. So just to recap, what Bliss and Lomo did were to make use of neural anatomy of the hippocampus, and then we talked about the perforant pathway, right, the inputs coming onto the dentate gyrus. If I can go into the slide, in the perforant pathway, these are the inputs from the entorhinal cortex EC, and they stick in an electrode, it is called as a stimulating electrode, and then they stick in a recording electrode in the cell body layer of the dentate gyral cells. If you remember, this is the first synapse in the hippocampus formation, and the argument is when something like an event can be thought of as a differential stimulation pattern. So what they were trying to do was that they wanted to look at the efficacy of these synapses, how efficient are these synapses, how much is the throughput of these synapses in response to a stimuli. The argument is, since they are globally measuring by sticking in an electrode near the cell body layer or in the cell body layer, this electrode will be measuring a sum total of all these responses. Now for a small stimuli that I am going to give here, I am going to look at what is the response. By that I can get a baseline level of what is the efficacy, what is the throughput. Then I am going to send in what the Bliss and Lomo are going to send in what they think or what they thought could be a plasticity inducing stimulus. Okay. We will come to the exact details of the stimulus in a minute, and then they are going to measure the same. The logic is, if by some means of changing the stimulus they can elicit a better reference, a better response or an improved synaptic efficacy in these synapses, we would like to ask how long do this efficacy stay after removing the stimulus. And that is exactly what they did. The way they did is in making use of rabbits, anesthetized rabbits, and they made use of the fact that we have two hemispheres in the brain. Mammalian brains have two hemispheres, and they are we could use one of the hemisphere for an experimental condition, while the other can be used for the control condition, wherein they would not be putting in the conditioning stimuli. Okay. What are these conditioning and the test stimuli? Now how did the experiment go? So having implanted these recording electrodes and the stimulating electrodes, what they did was to send in a test stimuli here. A test stimuli is a 0.5 hertz stimuli, that is a small stimulus that is sent once every 0.5 seconds, every 500 milliseconds, and then they are measuring the response at the recording electrode. Whatever and when you do that, what you will see is that a response something like that. So, now these potentials, this is an voltage response, this is a potentials and measured in voltage. This response is from the recording electrode, that is from the recording electrode. You can characterize an amplitude, you can characterize the rise time. In this case they are actually looking at the amplitude of the population response, and it is also called as a evoked potential, and you are measuring that potential as a and then normalizing it to be the 100%, and then the dots, individual dots measure tells you what is that amplitude for that one particular stimuli, that one particular pulse. You keep doing that for a brief period of time, you establish what is your baseline level, and they are at 100%, making sure they are not changing in time, it is very, very important. At this point in time where there is an arrow, they are giving a conditioning stimuli or a training stimuli, what as they call it, or a conditioning train of pulses, which is essentially the same stimuli, but then at a different frequency. They are now giving like 15 hertz. If one were to give simulate this, it is pretty much like initially they were going like tick 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 tick, and then for every tick they are actually measuring how much is the response. At this arrow they are going at 15 hertz. All of this pulses are given in a very brief period of time, and clearly right after that what you see is that the amplitude has gone up in the experimental group. In the control group, that is in the other hemisphere of the rabbit, they have not given that stimuli at all, and as a result they are same, and these are the amplitudes measured from the potential, and the potential per se you can see this is completely changed, right. That is the level, and they have increased in that amplitude, right, about. So, to get a measure, so that is the scale, right. So, that is about 2 millivolts scale and 10 millisecond in this axis, and you can clearly see they have really gone up compared to this dotted line or the dashed line. It is not just that it has gone up, but it stays flat. Now, you have stopped the stimuli here, right. You gave that brief stimuli and then stopped it, and you are continuously probing how good is the synapse, is it holding up, is it holding up that high voltage response, right. You are seeing that amplitude being high for a considerably long period of time. At this point again they give, and then they keep on repeating that, and it is continuously going up, and at this point in time they stopped giving any more stimuli at all, and what you see is there is a steady holding up of the increased amplitude in these synapses as against the controls, right. So, remember, so this differential enhancement in the efficacy of the throughput or the increase in the amplitude, they called it as potentiation, because the synapses are potentiated now, so that they can actually give in a higher response for the same amount of the input response, and that lasts. So, you call it as long term potentiation, that lasts much longer. So, it is about even 3 hours later. Now, there have been cases in the there have been labs which claim that they can hold this LTP for days, not just hours, even for days. Now, the beauty of this experiment is that this for the first time allowed us to even think of what could be a possible mechanisms through which a memory could be stored in the brain. Now, this does not mean memory is long term potentiation, but what it could be is that the mechanisms underlying the long term potentiation, that is the enhancement in the evoked potentials that you see in this experiment, could be same as that of one that happens when you form memory, right. So, granted it is a laboratory phenomena, the LTP, it is, I mean there is no necessary there is no equivalent necessarily for the outward, there is no equivalent stimuli in an outside world that is realistic, that can actually generate such kind of a response in the brain. But the fact is, when you do things like this, you are seeing an enhanced amplitude, and that stays, and your ability to, and it involves the neuron's ability to enhance or modulate its response and be able to hold that modulation for a long period of time. And the mechanisms involved in this could, I mean they have similar properties, similar time characteristics. So, it is tempting to correlate that they could be same as that of the one that are formed in memory. Now, this really opened up a huge field of study, because now you have a way of simply testing which are all the drugs, which are all the chemicals that can enhance LTP, that do they enhance memory, a plethora of, and you can talk about mechanisms that can be underlying this, what protein, what gene, what not. You can actually really dissect out, go ahead and dissect out various aspects of this regime, and in fact the field did expand and people did use this to the best part of then to now. As with any good things, there is always this issue of the debate about is LTP really memory. What I would like to say is that it is very highly correlated, that does not mean it reflects memory. Often this fails because people say that, hey look, there are several mechanisms which actually enhance or impair, which affect LTP, but that does not seem to necessarily affect memory. So, if it is really that, meaning this equals that, then that should be reflected in this correlation, that should really show that, right. So, to this we will talk about this particular review later. My good friend Young-Sok Lee, he posed the question in a slightly different way. Instead of asking the question of does all the factors that affect LTP, do they affect memory, he asked how many times have you seen a memory deficit or an enhancement without a change in LTP. It turns out that correlation is extremely tight than the other way around. Okay. Again alluding to the point that could be this gives us an opportunity to investigate possible mechanisms that may be common between LTP and memory, and it is that way it is a fantastic candidate, and it is probably one of the extremely highly studied candidates in the field of learning and memory, and for a good reason, as I was stating that. But however good the reason be, however good the candidate be, you would like to know what might be a possible reason why this is happening, right. What would be a possible molecular correlate. Now, in order to do that we need to understand a little bit. So, this is clearly happening at the synaptic level, and we need to understand a little bit more about the synaptic communication themselves. Now, if we look at the synapses here, adapting a picture from this famous book called Memory from Molecules to Mind by Candle and Square, a very good introductory prelude to anybody who is interested at the molecular details of memory and behavior to some extent. So, what happens is that as and when the nerve impulse or the action potential we talk about when you are talking to a neuroscientist, and whenever an action potential reaches the presynaptic bouton, in response to this action potential, the presynaptic bouton releases the neurotransmitters that are packaged in vesicles. So, these round entities that are shown here are vesicles that contain neurotransmitters. We have seen this neurotransmitter before, dopamine is one of them, but that is not the only one. There are many such neurotransmitters present in the brain, and one of the prominent one is glutamate. These neurotransmitters, glutamate for example here, then goes and binds to receptors that are present in the postsynaptic membrane, which depending on the kind of receptor it is can signal the postsynaptic membrane quite a few things, of which one particular thing being changing the conductivity of a class of ion channels or the class of receptors called the AMPA receptors. Here it is called non NMDA receptors, but we know that now the basic or a default conduction, neuronal conduction is taken care of by a set of receptors. What we call it as non NMDA here in this figure is actually AMPA receptors. They are actually receptors for the neurotransmitter glutamate, but you can differentiate them by using these drugs AMPA and NMDA, and they selectively bind to one fraction of the glutamatergic receptors, and the other one binds to the other. So, thereby you classify them into AMPA and NMDA receptors. That besides that point, what you have is there are two classes of receptors. I mean there are many classes, but in here we are looking at two classes of receptors, AMPA and NMDA. While AMPA takes care of the routine neuronal conduction, NMDA does a slightly different job. The job here is that it is a receptor for glutamate. At the same time it is also a sensor for an voltage. Now, normally when you look at these receptors, their conductance is blocked by binding of magnesium ion in its cavity. However, when the glutamate comes and binds to this receptor, and within a certain period you have an electrical activity in the postsynaptic neuron that releases this magnesium, thereby letting the calcium ions flow through this NMDA channels. Now, such a kind of a behavior, right, which is in contrast to the AMPA, right. AMPA we are talking about just a simple regular conduction, just ion channel, it is a simple ion channel, I mean simple receptor where it binds to the glutamate, and then you have the opening up of that pore, and then sodium and potassium exchange happens. So, neuronal conduction happens. However, in here for this pore to open, this receptor to open its ion channel activity, it needs to have two things. One, binding of glutamate, 2, sufficient electrical activity. At the postsynapse. Now, why would we be talking about this? Now, such a character, right, such a character of this receptor puts it in a very unique position. It can actually detect simultaneously the activity in the pre as well as the postsynaptic neuron, right. It detects the activity in the presynaptic neuron because it needs the glutamate to bind to it. It detects the activity in the postsynaptic neuron because without the electrical activity the magnesium would not be released, I mean without the electrical activity, and so it needs to be both that glutamate binding along with the postsynaptic activation opens up these receptors for neuronal conduction, and when it opens up, calcium ions being positively charged, they having higher concentration outside than the inside, they flow through these channels, thereby triggering a biochemical cascade. Now, again this activity of NMDA receptors puts them as a very high chance or very good candidates for detecting or coincident activity. They are called coincident detectors, thereby they have a ability to encode associative memory, and from that is the argument people from Susumu Tonagawa's lab used to generate a kind of a mice in which these receptors were absent. These receptors were absent specifically in CA1 region, all right. So, if you knock if you take away these genes from the mice, the resultant mice you generate, you call them as knockout mice, and you can generate these knockouts in various different ways. One of the ways is this is the region specific knockout of a given gene, and you can actually test them, is it region specific. Then in here what you are looking at as the images of expression of the mRNA. This is a stage before the receptors are proteins themselves. So, stage before the protein expression is the mRNA, presence of the mRNA. mRNA gets translated into the proteins, and you are looking at probing the levels of mRNA here, and the dark lines the black contrast here indicates the level of mRNA, and you can clearly see that in the control mice the NMDA receptors mRNAs for them are beautifully expressed and it is very high, while that is not so in here in the knockout mice. Okay. And it is specific to the CA1 region. You can actually go ahead and look at other regions, for example cortex or in DG or in CA3, they are all fine, they are not very different. Having generated this, they wanted to ask, now I am going to study the LTP, not the LTP the kind that I mean it is the kind that Bliss and Loma established, but in a different region. So, Bliss and Loma were looking at the DG to entorhinal cortex to the DG, right. They were looking at recording they were recording electrode at the cell body layer of the DG, but they are now here what we are going to look at is the LTP coming in or to the CA1 region, right. And then what do we see? If you look at the LTP, now the open symbols represent the control animals. There are there are 3 or 4 different controls, but does not matter. Let us not worry about the difference in the controls, but the idea is they are all controls, okay, for various different aspects. When you are doing a knockout study, it is important to establish quite a few things are not the reason why you are seeing this effect, and the bottom line here is you can do all those controls and it turns out they are all not the reason why you seeing this profound effect, wherein the open circles wherein you can see this the LTP in the knockout animals represented by the dark circles the filled circles is much lower, and in fact they do not have any LTP at all, they do not show any LTP at all. How do we know that? You see this response here, right, that is at the baseline level right after you stop, it comes back to the baseline level right after you stop this. It goes up a little bit, but then comes back down, okay, compared to the controls where it goes up and stays high at a much higher level than the baseline. So, having the NMDA receptors at least in this CA1 specific knockout in the CA1 pathway seems to be necessary for expressing LTP and inducing LTP and being able to maintain it. Now, we have granted we have not separated the idea of induction and the maintenance, but the point is that you need without this NMDA you are not going to have that LTP. So, there is no potentiation in the CA1 region without the NMDA long term potentiation without NMDA receptors being present there. Now, that is again it is a lab phenomena, right, that I said it is LTP is a lab phenomena and a laboratory induced phenomena. How relevant is it in terms of the behavior? So, clearly any experiment that one does to unravel the role of these different molecules in the behavior, particularly the memory and then related to LTP, need to test the memory too. And the way you do that is through a behavioral task. This is called as a water maze task. The water maze experiment involves taking an animal, rodent here, and then putting it inside a big pool of water, a big tub in which it is filled with the tub is filled with water, and you place the animal inside the tub. Rodents do not like to swim, but they are capable of swimming. They do not like to be in the water though. So, they would want to very first instance when they get an opportunity, they would like to come out of that water and stop swimming. And you put them in the pool, they will keep afloat by continuously swimming, but inside that you hide a platform. What do I mean by hiding? You place a platform just about the level of the water. So, if you are at the level of the water, you will not be able to see it. Added to the fact, the water itself is also opaque. You make them opaque, make the water opaque by adding milk powder or some turbidity inducing substance. As a result, you will not be able to see through the platform. So, you do not know where the platform is. However, the rodent have to figure out where the platform is, that is the task. So, you train them over repeated trials to learn where the platform is. So, how would it learn? What are the cues? The cues are placed as objects present outside the maze. These are extra maze cues that are placed on the room around the tub, and using that cues it has to figure out where that platform is. Not just that, it has to figure out and remember, so that the next time when it comes back it can go to that same place. Now, the mice take some time to learn this task, and you can measure the performance, the progression in this task by asking how long a time the mice takes before it actually reaches the platform. And if you do that, that is called as an escape latency, or the latency to actually reach that platform. And when you measure that, you will see a continuous progress, and then latency comes down. And you can also look at trace its path, because you are recording through the video. So, you can actually trace its path. Initially it does not have any clue, it is circling around all over the place. Over a period of time it develops a strategy and develops an association between the cues and the position of the platform, thereby slowly refining its path such that towards the end where it really has learned the task, you can actually reach that platform in a very quick time. Again, whenever you are testing memory, you actually want to delineate the training, which largely involves the learning aspect, from the probing or the testing, which actually which largely involves the memory aspect. See, both the training and the testing involves a combination of learning and memory. It is never only one or the other, but what we are talking about here is we bias such that the training is dominantly learning and testing is dominantly memory retrieval. Okay. So, now what we are doing here is that we talked about the training phase here, and then how do we probe the performance. We want to know how do we actually test the memory capacity. The way you do that is by doing a probe test, right, and just in our traditional CS US experiments, we would remove the US and ask what is the response for the CS, right. Same way here, what we will do is we will remove the platform and then say what is the mice doing now. If the mice is actually understanding or remembers that this is where the platform is, then it will selectively search a larger fraction of the time that is given to it in the place around the where the platform is. And you can measure it. You measure it by dividing this entire circular pool in 4 quadrants and measuring what fraction of the time the mice spent in each of these quadrants. In short, when you do this, it turns out that if the animal has learned, it will end up spending a large amount of time in the platform quadrant. It is also called as a target quadrant. Now, this behavior depends on hippocampus. How do you know it? You can lesion it out. If you lesion it out, what happens is that the mice searches all around the quadrants. It is just moving around all around, and it is not that it can't find it at all. You have to understand that for it to find, you need to really overtrain them, but the point is at a moderate, at the regular training, it needs if you lesion out the hippocampus, it can't find the platform. In the next lecture, we will see how this group use this behavior to test the requirement of NMDA receptors in the CA1 region in forming the memories for this task. Thank you.